So, hello everyone. My name is Masoud Amasian. I'm chair of ENIGMA-Sleep Consortium, and I'm very happy to introduce the speaker of 16th ENIGMA-Sleep talk, Dr. Claire André. Claire obtained her PhD from Caen and Normandy, if I'm not mistaken, University in France. Then she did her postdoc in Montreal, and now she's back in France and working in the research institute, I guess. So her main focus is on the neural association between sleep and biomarkers of Alzheimer disease. I think she will explain some of like her research, her recent findings. And we'll have like 15 minutes to discuss them at the end of the talk. Thank you, Claire, for accepting my invitation, and the stage is yours. Thanks.
Thank you very much for the introduction and the invitation. It really is a pleasure to be presenting some of my recent work on sleep and Alzheimer's disease today. So I'm going to start with a very short introduction because I'm pretty sure you are all quite familiar with this already, but you know that sleep disturbances are very frequent and severe in patients with Alzheimer's disease, especially when they suffer from dementia, so in quite advanced stages of the disease. The type of disturbances that we see is pretty much an exaggeration of what is observed in normal aging. So the duration of sleep is reduced, sleep is more fragmented. We also see an impairment of slow wave sleep, which is reduced, and we also have a decrease in slow wave activity, so slow oscillations are impaired. We do see a higher prevalence of sleep disorders, including obstructive sleep apnea, that I'm going to talk about a little bit later. And we also see more specific changes, and this is the case of REM sleep alterations, which are not normally observed in normal aging. These sleep disturbances occur quite early in the course of the disease, and they are also associated with the progression of the disease. So, for example, in epidemiological studies and longitudinal studies, we see that people reporting sleep disturbances are more likely to develop dementia in the future. And you see here an example of a recent meta-analysis showing in participants who had a mean follow-up of approximately 10 years, that those with sleep disturbances have a higher risk of developing dementia, and especially Alzheimer's disease. And it's especially the case for those reporting sleep apnea, but also all cause of sleep disturbances. So this led to the idea that sleep problems could represent a risk factor for dementia, but we still need to progress in our understanding of the underlying brain mechanisms and how sleep disturbances are associated with underlying Alzheimer's disease pathology, so amyloid, tau, and neurodegeneration, especially in early stages of the disease. And the field has been working quite intensely in these recent years. You can see here the increase in the number of papers published on the topic of sleep and Alzheimer's disease in PubMed. In 2023, we had more than 600 papers published on this topic compared to around 80 papers 10 years ago. So the field is really growing exponentially. And we are progressing on our understanding on which aspects of sleep are associated with Alzheimer's disease pathology and symptoms. And a few sleep aspects that appear to be really important is first, obstructive sleep apnea. We are understanding more and more the negative impact of untreated obstructive sleep apnea on brain health. And I'm going to present you some work that I've been doing on the topic. We also understand more and more the importance of non-REM sleep integrity, which supports memory consolidation and lymphatic clearance. And I'm not going to talk about this today because I've seen that you already had excellent talks and multiple talks on the topic, so you are already experts on the matter. And some aspects of sleep are more understudied, such as REM sleep alterations and circadian rhythm. And I'm going to talk especially about REM sleep today. So we're going to start with untreated obstructive sleep apnea. So you probably know that obstructive sleep apnea is a sleep disorder that consists in total or partial interruptions of respiration during sleep. And this has negative consequences because it provokes oxygen desaturation, and it leads to intermittent hypoxia, which is harmful for the brain. And also, it is associated with arousals on the EEG. So this is causing sleep fragmentation on the long term. So obstructive sleep apnea is very prevalent in older people because we see that up to 50% of older adults may suffer from obstructive sleep apnea, and this percentage is higher in some subgroups, and especially it's higher in people with dementia. And untreated sleep apnea is associated with cognitive decline and an increased risk of developing dementia in epidemiological studies. But we need to understand more why this is the case and what can possibly be the underlying brain mechanisms. And this is what I've been interested in my PhD in France. This is a work that we've been doing on the French age-well cohort in Caen. We had 127 cognitively unimpaired older people. They all had a neuropsychological evaluation just to ensure that they were cognitively normal. They had a polysomnography from which we extracted respiratory features. And they also had multimodal neuroimaging. So we did a structural MRI to assess gray matter volume. They also had a floor beta PET scan to assess perfusion in an early acquisition and amyloid deposition in a late acquisition. And a subset also had an FDG PET, which is measuring glucose metabolism. And the first thing that we've been doing is quite simple, actually. We wanted to compare the profile of brain alterations between participants without sleep apnea, so those presenting with an apnea-hypopnea index below 15 events per hour. And we had 31 participants without sleep apnea, which is less than what we expected. And compared to those with moderate to severe sleep apnea. And we had 96 participants with moderate to severe sleep apnea. And what we observed when we compared the two groups is that people with moderate to severe sleep apnea had greater amyloid deposition in posterior cortical areas. And in the same areas, they also had a higher gray matter volume, higher perfusion, and higher glucose metabolism. So it was really the most significant in parieto-occipital areas, so the cuneus, precuneus, and posterior cingulate cortex, which are sensitive to Alzheimer's disease. And the overlap between these patterns of brain alterations between the modalities which were independent was quite substantial. So we were suspecting common underlying mechanisms here. What we did next was that we wanted to clarify which aspects of sleep apnea was the most closely related to brain alteration and the pattern of brain alterations that we were seeing. So we did forward stepwise regression analysis, and we assessed whether the apnea-hypopnea index, so the number of respiratory events and their frequency, basically. Intermittent hypoxia parameters or sleep fragmentation parameters were the most closely associated to amyloid deposition, which was the larger cluster. And actually, hypoxia was the parameter most closely related to amyloid deposition. So it seems that intermittent hypoxia is really the aspects of sleep apnea that was harmful for brain health here. And this is quite interesting, but we had no association with cognitive performance. All the participants had a neuropsychological evaluation, which was quite extensive. It covered attentional processes, episodic memory, basically everything, executive functions. And we had no association with any cognitive alteration and cognitive performance. So all the brain alterations that we saw were cognitively asymptomatic. So this is a quick summary of the results of this first study. We observed that sleep apnea and especially hypoxia was associated with higher gray matter volume, perfusion, and metabolism in these posterior cortical areas. And we had several hypotheses of why this is the case. It could reflect neuronal hyperactivity, for example, but also neuroinflammatory processes and reactive gliosis or cerebral edema. So this is what we observe in animal models, and this could perfectly underlie these patterns of higher gray matter volume and metabolism. And this could increase the accumulation and the production of amyloid deposition, which could, of course, on the long term be associated with cognitive decline. We did also a follow-up study just to dive a little bit more and verify this model. In a second study, we were interested in assessing what happens once participants become amyloid positive. So in later stages, right here, if sleep apnea is promoting the accumulation of amyloid deposition, we were interested to see whether later in the course of the disease, sleep apnea could also be associated with neurodegeneration and cognitive decline. So we separated amyloid positive and negative participants, and we compared them and assessed the associations with neurodegeneration. So this work was done in the same cohort. We had 122 cognitively unimpaired older participants. They had the same set of examinations, of course, at baseline. And we were here interested in comparing the differential effects between participants who were amyloid positive and negative. And we had 26 amyloid positive participants. And we also had a special interest regarding structural MRI data on hippocampal subfields and subregions of the medial temporal lobe, because when you read the literature, you see that these regions are quite sensitive to hypoxia. So we wanted to check whether sleep apnea had an effect on medial temporal lobe subregions. And these participants were followed. They had a cognitive follow-up approximately 21 months after. And at follow-up, we had a sample size of 111 participants. So first, for cross-sectional analysis, so analysis at baseline, we observed that greater sleep apnea severity, so a higher apnea-hypopnea index and a higher oxygen desaturation index, so a more frequent breathing events and more severe hypoxia were associated with a reduction of the volume of the hippocampus here. And some specific hippocampal subfields, so the subiculum, CA1, and dentate gyrus subfields, and also the entorhinal cortex. And this was only significant in amyloid positive individuals. So it means that in amyloid negative individuals, sleep apnea had no impact on, was not significantly associated with gray matter volume in any of these medial temporal lobe subregions. We checked whether this pattern was really restricted to medial temporal lobe subregions. And it was the case. We did voxel-wise analysis with global T1 images. And we saw that the associations between the apnea-hypopnea index and hypoxia metrics was quite restricted to medial and lateral temporal areas in amyloid positive individuals. Something really interesting is that it was driven by women in sex stratified analysis. So it means that all the significant associations that you see here were significant in women, but not in men when we separated men and women. And we had very, very marginal associations with microarousals metrics, so sleep fragmentation. So again, it was really hypoxia which was the metric driving the results. We next assessed whether we had significant associations with episodic memory at baseline, so cross-sectionally and also longitudinally at follow-up. We had no significant associations at baseline, so it kind of mirrored what we had in the first study. But when we looked at longitudinal performance, we had in the whole cohort a significant association between lower baseline hippocampal and CA1 subfield volume and lower episodic memory performance at follow-up. So baseline hippocampal and CA1 volume were significantly predicting episodic memory performance at follow-up 21 months later. So it means that sleep apnea could promote amyloid accumulation, and once individuals become amyloid positive, sleep apnea could then be associated with neurodegeneration, especially in the medial temporal lobe. It was quite restricted in our cohort. I suspect that it's because the participants are cognitively unimpaired, so quite early, of course, in the course of the disease. And the medial temporal lobe is one of the first cortical regions to be affected by Alzheimer's disease pathology, so it makes sense. And it could promote long-term memory decline. So this is where my PhD ends. So this is some work that I've been doing during my PhD, but then I moved to Montreal to start my postdoc. So I worked on a different topic, but I continued to collaborate in Montreal on studies on obstructive sleep apnea, and it really makes sense regarding the model that we wanted to investigate here, because we did a study in Montreal in order to understand a bit more why we see increases in gray matter volume in participants with obstructive sleep apnea. So we wanted to check whether these gray matter volume increases reflect reactive processes like, for example, edema or neuroinflammation. So this was a work conducted by Marie-Ève Martineau-Dussault, who was a PhD student at the time, on the Montreal cohort. So we had 123 participants aged between 55 and 90 years old. And they basically had the same set of examination that in the French cohort, but we were here interested in verifying whether the increases in medial temporal lobe volume that we see in association with obstructive sleep apnea were affected by changes in brain water content by applying a free water correction to gray matter volume. So we derived free water values from diffusion images, and we corrected our gray matter volume by this value. And It's really interesting because in raw gray matter volume, we saw the same type of effects in the Montreal cohort compared to the French cohort. So here we saw that increased sleep apnea severity was associated with greater gray matter volume in the hippocampus and entorhinal cortex. So this was especially significant in women, again, and not in men, but it was also significant in other groups. So older participants with MCI, for example, but this is an example with a woman. And when we controlled our volume by the amount of brain water, so free water values, these associations were no longer significant. So sleep apnea was no longer associated with gray matter volume increases. So it really supports the fact that these increases are not gray matter volume increases per se, but are reflecting increased brain water content, so edema. So these increases in gray matter volume that we see quite early in reaction to obstructive sleep apnea may be maladaptive and reflects acute increases in brain matter content and edema. And we also wanted to check whether beyond structural alterations, we had functional connectivity alterations consecutive to obstructive sleep apnea. And this is a study that I supervised by Guillermo Martinez and Véronique Dano on the same cohort in Montreal. We performed ROI-based analysis on resting state fMRI data between brain regions that are known to be sensitive to Alzheimer's disease. And this is what we observed. We observed that a higher sleep apnea severity was associated with lower functional connectivity between the hippocampus and the medial prefrontal cortex, and the hippocampus and posterior cortical areas. So this is where I had a greater accumulation of amyloid deposition in my first study. And these associations were driven by cognitively unimpaired participants, so quite early in the course of the disease. So just to summarize the work we've been doing on obstructive sleep apnea, untreated obstructive sleep apnea is associated with structural, functional, and molecular brain changes that are suggestive of Alzheimer's disease. So we see first acute reactive gray matter changes and higher amyloid levels. And quite later, it's not very late, but quite later, we could see neurodegeneration appear and lower resting state functional connectivity, as well as greater tau levels. We did not have tau measures, but other groups have shown higher tau accumulation, specifically in medial temporal lobe and medial temporal areas. Taken together, this may exacerbate a long-term cognitive decline, especially because the medial temporal lobe is crucial for memory processes. And it leads to a follow-up question, which is, can CPAP treatment reverse or at least slow the negative consequences of obstructive sleep apnea? And it seems quite promising because this study by the Italian group of Claudio Ligori showed that actually participants that are treated for obstructive sleep apnea exhibit the same level of Alzheimer's disease pathology than controls compared to those who were not treated, who show higher levels of amyloid pathology. So it seems quite promising and supports the fact that possibly if you detect and diagnose quite early obstructive sleep apnea, you could prevent brain alterations. And another aspects of sleep that I'm really interested in is REM sleep. So I'm going to present you some of my postdoc work on REM sleep. It's really an interesting and intriguing sleep stage because a lot has been done on non-REM sleep, but not a lot on REM sleep. And we should really improve our understanding on REM sleep because REM sleep is really impaired in patients with Alzheimer's disease, but it's relatively preserved in normal aging. And in patients with Alzheimer's disease, we see a reduction of REM sleep duration and a global slowing of EEG rhythms during REM sleep, which is not supposed to happen. REM sleep is a sleep stage during which cortical activation is really intense. These changes are, again, detectable before the onset of dementia in individuals at greater risk of Alzheimer's disease. This is the case in people with mild cognitive impairment or people who carry the APOE4 carriers who are at greater risk of developing dementia. And lower REM sleep percentage, so lower REM sleep duration, has been associated with incident dementia, even when you adjust for multiple confounders, such as vascular risk factors, depressive symptoms, or medication use. So REM sleep disturbances could really represent an early marker of underlying AD pathology, but this needs to be verified. And this is what I've been working on quite recently. First, one last study of my PhD on the French age-well cohort. We had 121 cognitively unimpaired participants, same cohort than before. They had the neuropsychological evaluation, the polysomnography examination at home. And here we were interested in REM sleep duration and REM sleep spectral power in standard frequency bands. And they, again, also had multimodal neuroimaging, so structural MRI, floor beta PETs, and FDG PETs. We showed in this first study that lower theta power was associated with greater amyloid levels. This is the voxel-wise associations between theta power and amyloid deposition. So lower theta power was associated with quite a diffuse pattern of increased amyloid deposition in the neocortex. And when we compared amyloid positive and negative individuals, the only difference that we saw in terms of sleep was, again, lower theta power in amyloid positive individuals. So theta power during REM sleep was quite strongly associated with greater amyloid deposition. But in this study, we had no major association between REM sleep integrity and cortical gray matter volume. This was quite intriguing at first, and I wanted to investigate this a little bit more during my postdoc. And during my postdoc, I was really interested in the associations between REM sleep and the cholinergic system. Because subcortical nuclei in the brainstem and basal forebrain are affected by Alzheimer's disease pathology and especially tau pathology before medial temporal lobe regions and the neocortex, of course. And we notably see an early atrophy of the cholinergic basal forebrain, so the nucleus basalis of Maynard's, which is, by the way, the main source of cholinergic cortical neurons in the neocortex. And something really interesting is that REM sleep, and notably the cortical activation, which is really intense during REM sleep, are really dependent on cholinergic activity. And cholinergic activity is maximal during REM sleep. So this high pattern of cortical activation is really cholinergic dependent and typical of REM sleep. So the main topic that we have been working on is clarifying the associations between sleep disturbances, REM sleep disturbances, and the disruption of the cholinergic system in older populations and patients with MCI. So we did a first study in which we assessed the structural integrity of the basal forebrain and its associations with REM sleep. So we had a sample of 62 participants. This is quite a restricted sample because we excluded everyone with significant levels of obstructive sleep apnea during REM sleep. So we had to discard in this specific analysis 56 participants of our sample because they had moderate to severe levels of sleep apnea during REM sleep specifically, which was a confounding factor here. And in this sample of 62 participants, we had 31 cognitively unimpaired individuals and 31 participants with amnestic MCI, so prodromal Alzheimer's disease. They did not differ in terms of age, sex ratio, and level of education. They had polysomnography in the lab, and we extracted REM sleep architecture metrics, so REM sleep duration, latency, and efficiency. And they also had a structural MRI, and we extracted the volume of basal forebrain subregions. And this is what we observed. We saw that lower REM sleep percentage was associated with a lower volume of the basal forebrain, and especially of the nucleus basalis of Maynard. This was not the case with the CH123, which are the medial septal and diagonal band of Broca nuclei. So it was really specific to the nucleus basalis of Maynard, and only significant in participants with amnestic MCI, so prodromal Alzheimer's disease. And it was also significant when we average all the nuclei together. And something really important is that we had no significant association between basal forebrain volume and the duration of wake after sleep onset or non-REM sleep stages. So the associations between basal forebrain volume and REM sleep were specific to REM sleep, actually. But this is structural imaging, and in the basal forebrain, there is no, it's not specific to cholinergic neurons. You also have other types of neurons. So we wanted to next assess the integrity of the cholinergic system more specifically, and to do so, we did PET imaging with the FEOBV tracer, which is a tracer showing a high affinity and specificity for the vesicular acetylcholine transporter, and it reflects presynaptic cholinergic density very specifically. And previous study by other groups have shown that cholinergic uptake is reduced in Alzheimer's disease and MCI patients, and it also correlates with the magnitude of basal forebrain atrophy. So the tracer is working well, and we do see a cholinergic deficits in patients with MCI and Alzheimer's disease. But no one has tested the associations with sleep before. So this is what we did. So this paper is not published yet, but it's on its way. We had 24 older participants with an age range between 65 and 80 years old, quite well balanced in terms of sex ratio. And something really important is that they did not have major comorbidities, including obstructive sleep apnea again. We had quite a restrictive sample because the tracer was considered experimental at the time, so we were restricted. They had a neuropsychological evaluation, so we had 18 cognitively unimpaired participants and 6 patients with amnestic MCI. They had polysomnography in the lab, and we extracted REM sleep metrics. So the percentage of REM sleep theta power, and the global slowing of EEG rhythms during REM sleep. And they had a structural MRI and cholinergic PET examination. The main thing that we did was to assess whole brain voxel-wise associations between cholinergic maps and REM sleep characteristics. We controlled all the analysis for sex as a first step, because we observed that cholinergic uptake was higher in women than in men, especially in temporal parietal areas. This is what you can see here. But we also confirmed the main results that we had in models adjusted for age, sex, and cognitive status. So this is our main observation. We show that greater REM sleep EEG slowing over frontal derivations and parietal derivations was associated with cortical cholinergic denervation. And this is quite a diffuse neocortical pattern. It's maximal in frontal and parietal areas, as well as in the medial temporal lobe, but still it's quite diffuse. You can see here, it's quite closely associated. So the higher cholinergic uptake, the lower the slowing ratio is, which is expected. So when you have a cholinergic denervation, you have here a higher slowing ratio, so less cortical activation. It was still significant in models adjusted for age, sex, and cognitive status. We, again, had no significant associations with EEG slowing ratio computed during non-REM sleep, so N2 and N3, taken together, as well as resting state wakefulness. The participants also had a resting state wakefulness EEG assessment, and we computed the slowing ratio specifically on resting state EEG, and it was not associated with cholinergic denervation. So again, this was specific to REM sleep. We also had no significant associations with other REM sleep variables. And one of the next steps will be to assess in longitudinal studies whether REM sleep has a prognostic utility to predict cognitive decline and dementia. So just to sum up, on the work we have been doing on REM sleep, we show that REM sleep disturbances are emerging as a potential marker of underlying Alzheimer's disease pathology, so greater amyloid levels, as well as cholinergic denervation. But there is still a few questions that we need to answer. So what is the impact of tau pathology, and what are the clinical consequences of REM sleep disturbances? And this is what I'm investigating right now. So with an ongoing project on Prevent AD data in Montreal, we are interested in investigating the impact of tau pathology on the associations between sleep-wake disturbances and the integrity of the cholinergic system. Because tau, as I said, is really affecting subcortical nuclei quite early in the course of Alzheimer's disease. And also, we are now in France and also in collaboration with Quebec, investigating the impact of sleep disturbances on the emergence of neuropsychiatric symptoms in aging and neurodegenerative diseases, because especially REM sleep, but sleep is not only important for memory and cognitive processes, but it's also important for emotions regulation, and could be associated with the emergence of neuropsychiatric symptoms. So this is some future directions that we're investigating. So yes, that's all for me today. So I want to thank all the people that have been involved in all the studies that I've presented, and in particular my postdoc mentors, Nadia Gosselin, Julie Carrier, my PhD mentors, Géraldine Roche, Gaëlle Chetla, and also my collaborators right now, André Baril and Sylvia Villeneuve. And thank you very much for, again, the invitation and your attention.